Good afternoon, everyone. My name is Laura Morell, and I work in the National Center for Emerging and Zoonotic Infectious Diseases at the Centers for Disease Control and Prevention. On behalf of the One Health Office, I'm pleased to welcome you to the monthly Zoonoses and One Health Updates call on August 4th, 2021. Next slide, please. Although the content of this webinar is directed to veterinarians, physicians, epidemiologists, and related public health professionals in federal, state, and local positions, the CDC has no control over who participates. Therefore, please exercise discretion on sensitive content and material, as confidentiality cannot be guaranteed. Today's webinar is being recorded. If you have any objections, you may disconnect now. Next slide. Today's presentations will address one or more of the following 5 objectives. Describe 2 key points from each presentation. Describe how a multi-sectoral One Health approach can be applied to the presentation topics. Identify an implication for animal and human health. Identify a One Health approach strategy for prevention, detection, or response to public health threats. And identify 2 new resources from CDC partners. Next slide. In compliance with continuing education requirements, all presenters must disclose any financial or other associations with the manufacturers of commercial products, suppliers of commercial services, or commercial supporters, as well as any use of unlabeled products or products under investigational use. CDC, our planners, presenters, and their spouses or partners wish to disclose they have no financial interests or other relationships with the manufacturers of commercial products, suppliers of commercial services, or commercial supporters. The Planning Committee reviewed content to ensure there is no bias. The presentations will not include any discussion of the unlabeled use of a product or a product under investigational use. CDC did not accept commercial support for this activity. Next slide. Instructions for receiving free continuing education are available at cdc.gov/onehealth/ZOHU/continuing education. The course access code is OneHealth 2021. To receive free CE for today's webcast, complete the evaluation at cdc.gov/TCE online by September 6th, 2021. A captioned video of today's webinar will be posted at cdc.gov/onehealth/2021/august.html within 30 days. To receive free CE for the web on demand video of today's webinar, complete the evaluation at cdc.gov/TCE online by September 7th, 2023. Next slide. Before we begin today's presentations, Dr. Casey Barton-Baravesh, Director of CDC's One Health Office, will share some news and updates. You can begin when you're ready.
Hi, everyone, and thanks for joining us on today's ZOHU call. We really appreciate you being here today. Before our presentations, I want to share a few One Health COVID-19 updates and highlights from today's ZOHU call email newsletter. And if you're not yet subscribed, you can use the link at the top of the main ZOHU call webpage so you can stay informed. CDC's response to the COVID-19 pandemic continues to evolve. Please check CDC's website for the latest guidance and resources, including information about keeping people as well as pets and other animals safe and healthy. We also hold a regular One Health Partners COVID-19 webinar where we provide news, key updates, guidance, and resources for public health officials, animal health officials, veterinarians, industry and academic partners, pet owners, and others. And you can email onehealth@cdc.gov if you want to get updates and information on how to join the webinar as well as the call schedule. All right, next slide, please. There's no evidence that animals are playing a significant role in spreading COVID-19 to people, but we do continue to see animals reported with SARS-CoV-2. In the United States, there are 218 animals from 29 states that have been reported, and these include companion animals like cats and dogs, large cats in zoos and sanctuaries, otters, gorillas, and mink. And there are now 17 mink farms that have been affected by SARS-CoV-2. You can find the latest animal case numbers available on the USDA APHIS website shown here. And there's guidance for pet owners, mink farmers, veterinarians, and many others available on CDC's website. Next slide. We wanted to highlight some recent publications of interest, including recognition of diagnostic gaps for laboratory diagnosis of fungal disease with expert opinion from the Fungal Diagnostic Laboratories Consortium. Also, the role of interspecies recombinations in the evolution of antibiotic-resistant pneumococcus. Knowledge gaps in the epidemiology of severe dengue and ped vaccine evaluation. Next slide. Some new web resources of interest. We wanted to make you aware that CDC has a ZOHU Call YouTube playlist available, and there's information out on how to apply for a CDC dog import permit, as well as some others listed here. Next slide. In terms of upcoming events, we have a long list here and wanted to highlight a few. August is Immunization Awareness Month, and the International Conference on Emerging Infectious Diseases, or ICEID, abstract submission deadline is October 1st. Next slide. And finally, some information on ongoing outbreak investigations. There's a new E. coli outbreak linked to cake mix, 3 new salmonella outbreaks linked to packaged salad greens, frozen cooked shrimp, and raw frozen breaded stuffed chicken products. There's a new listeria outbreak linked to fully cooked chicken. And as always, you can visit CDC's Healthy Pets, Healthy People website for a selected list of ongoing and past US outbreaks of zoonotic diseases. Our next ZOHU call is going to take place on September 1st. And we welcome you to email topic suggestions for future presentations and news from your organization, and even to volunteer to present on your work. We appreciate your help in spreading the word about the ZOHU call through sharing the web link with your colleagues from human, animal, plant, environment, and other relevant sectors, and letting them know about the live webinars, as well as free continuing education and video recordings of past webinars. Now I'll turn the call back over to Laura. Thank you.
Thank you, Dr. Barton-Barabash. Next slide, please. You can submit questions at any time using Zoom's Q&A feature. Please include the topic or presenter's name with your question, and the Q&A session will follow the final presentation if time permits. You can also email questions to today's presenters. We've included their email addresses on this slide, on the ZOHU Call webpage for today's webinar, and in today's email newsletter. Next slide, please. Our first presentation, human-to-human cattle transmission of Mycobacterium tuberculosis complex species, is by Dr. Jason Lombard. Please begin when you're ready.
Thank you, Lauren. Thank you for the opportunity to present on the ZOHU call today. I'm a veterinarian with USDA's Veterinary Services Field Epi Investigation Services, and it's my pleasure to talk to you today about human-to-cattle transmission, of which M. bovis and M. tuberculosis are both part of that complex. Next slide. So we'll start with cattle to human transmission, and we're all familiar with this. Cattle are most commonly infected with Mycobacterium bovis. As we all know, it's a zoonotic disease, and the cases of Mycobacterium bovis in humans in the early part of the 1900s led to the formation of the Cooperative State Federal U.S. Bovine TB Eradication Program in 1917. And that program has been very effective, yet we tend to find, and this is a typo, about 2 to 12 newly infected herds annually. And the source is often undetermined in those herds. We don't know why these herds, how these herds are getting infected, even after intense epi investigations looking at all the incoming cattle, potential fence line contacts, and other sources that we've looked at over the years. Next slide. In terms of human disease, this graphic that's put out by a number of well-known organizations, including the World Organization for Animal Health, pretty clearly defines how people are exposed. And on the left side, infected wildlife, primarily through hunters, is one way people can get zoonotic TB. But the more common route is probably through cattle on the right side of the figure, and that's through the consumption of unpasteurized dairy products or raw or improperly cooked meat, and potentially direct contact with those animals. Next slide. So since the program began in 1917, where about 5% of the animals responded to the skin test, In the 1940s, we dropped well below 1%. And in 2017, it's estimated that about 0.1% of the animals are responding to the skin test, to just show that the program has made a lot of progress in terms of removing infected animals, with the skin test being kind of the proxy for that. Next slide, please. This is a figure that shows the number of new cases of TB each federal fiscal year going back to 1900, or 1990, excuse me. And you can see that the trend line that with the dots starts at about 6 and has a slightly upward trend to about 7 herds per year. So it's been relatively stable over that time period, but we still continue to find newly infected herds each year. Next slide. So if we switch and talk about human to cattle transmission, and there's been 3 fairly recent US bovine TB cases that I want to discuss with you today. Next slide. The first case is M. bovis, detected in a North Dakota dairy herd. There was a dairy worker who had been with the dairy for many years who was diagnosed with TB disease. Public health alerted animal health that they had a dairy worker with TB, and they worked with the producer to get the cattle tested. At that time, there were 3 cows that were confirmed infected with M. bovis, and one of those cows was an exact match to the human case. Now, the human had severe TB disease, and the affected cow had a very small lesion in one of her lymph nodes. So it was, and based on the matching of the whole genome sequencing, it was determined by the folks in North Dakota that the human was the source of the cow case. That dairy did enter a test and remove protocol where they went through annual testing, and no additional infected cattle were affected. Next. So this is what we call a SNP table. So it shows which nucleotides are in which position in the genome. Next. So in the box here are the 3 isolates that are associated with this North Dakota herd. The first isolate is the dairy cow, and you can see that those what we call single nucleotide polymorphisms, or the colored boxes, match exactly to the human on the second row within that box. The third row lists the second infected cow, which had a slightly different genotype than the first 2, and there's some speculation on what may be going on there. The third cow that was confirmed infected with M. bovis was only positive by PCR. No culture was able to be performed, and so we only have those 2 isolates from that case. Next. The second case is again M. bovis in a Wisconsin dairy. This time we have a dairy worker who had been on the farm for 2 to 3 months that was diagnosed with severe TB disease. Again, animal health worked with public health through the notification of public health, and those cattle were tested. They had 2 negative herd tests at that time, and based on consultation with national staff, that herd was released from quarantine. 3 years later, an infected cow was found at slaughter that traced back to this Wisconsin dairy, and the whole genome sequencing matched the human case, with the only difference is that all the cows had an additional single nucleotide polymorphism than the human. That dairy also entered a test and remove protocol, which is ongoing with 12 identified infected cows to date. Next. And this is the SNP table from that Wisconsin herd. The very top one is our root sequence, and then the second line is the human. And we can see over here on the right side, there's an adenine in this location, and that's a guanine in all of the cattle isolates that have been detected so far. So we know that once a single nucleotide polymorphism is developed, those usually never revert back. And so by all evidence we have here is that the human did transmit this to the cattle, and that's pretty well backed up by the fact that the worker had disease and the tests were negative initially, and then it took a period of time before disease actually progressed in that herd to where it was detected. Next slide. This case is a M. tuberculosis in a Texas calf, not in a herd actually. It was a dairy crossbred calf that was tested because of movement from New Mexico into Texas based on Texas regulations. And the calf was test positive, was necropsied, and M. tuberculosis was cultured. As we all know that more than 95% of all human cases of TB are caused by M. tuberculosis, and it's not very commonly found in cattle. No human patient was identified associated directly with this calf. No employee testing was conducted on the operation. According to public health, though, there was a calf isolate. The calf isolate did match a human TB strain in the local area, but for some reason, those data were never put together to compare exactly the genomes of the calf and those patients. Next. This is just the tree of M. tuberculosis. The calf is highlighted in red, and within the database, the closest ancestor we have to that is a patient from the Netherlands. Next slide. All right, so we had the opportunity to publish these 3 cases just last month in the Frontiers in Veterinary Science, and I really want to thank the co-authors that did most of the work on this paper and on these 3 cases. So Dr. Patton, Dr. Gibbons-Bergener, Drs. Klaas, Tans-Kernsten, Carlson, Keller, Pritchett, Rollo, Dutcher, Young, Hench, Thacker, Perea, Lemkool, and Rob Osterman. Thank you. They all deserve credit for this presentation as well. Next slide. So those were US cases. What about other countries? And if we look over at the US, I've got a 2 there, and that's because there were 2 other reports in the literature, one from the 1940s and one from the 60s, that reported where a herd owner had potentially infected their own cattle. They'd been infected by their cattle. Those herds had been depopulated, and then it was presumed that they infected their new herds that they had brought in. Unfortunately, there's not a lot of good laboratory evidence to confirm that those were the same M. bovis strains, but interesting cases. The continent, actually, Africa has got quite a few of those, and that's cases. You can see 6 in Ethiopia, for instance, and those cattle are raised in a lot different environments than we have in the US, and so there's more direct contact and more opportunity for spread of Mycobacterium tuberculosis complex organisms. India has 5, China 2, New Zealand even has 2 out here, and then some in the EU. And these are actually the number of published articles, not the number of infected animals. A lot of these manuscripts report on hundreds of animals potentially being infected, cattle being infected by humans. Next slide. This figure shows a number of published studies by MTBC species and by the decade published, and you can see that the 1940 M. bovis, so M. bovis is denoted by the orange, M. tuberculosis by the blue. The 1940 and 1960 M. bovis cases are in here. We don't see a lot of M. bovis reported throughout the decades. In 2010, we had 17 articles that reported M. tuberculosis transmission from humans to cattle. As far as cattle to cattle transmission of M. tuberculosis, there's only been 1 report, and that was in an experimentally infected cow that transmitted M. tuberculosis to her calf. Otherwise, cattle, at least at this point, appear to be a dead-end host for M. tuberculosis. And only 1 case of M. bovis, and I think that's partially reflective of the fact that like I mentioned earlier, 95% of people with tuberculosis, with TB, have M. tuberculosis. Next slide. So as we go back to this graphic developed by the World Organization of Animal Health and others, I think we probably need to put a bidirectional arrow in here between people and cattle, because we do have a lot of evidence that's accumulated worldwide over the last few decades that there is the possibility of transmission from humans to cattle. Next slide. So in conclusion, the US continues to see cattle herds that become infected with M. bovis. Next. The source is really unknown for about 40% of those. And like I mentioned earlier, there's intense investigations that are done to try to determine which cattle have been added to that herd that might have brought in the disease, whether they had fence line contact or some other contact with infected cattle. Of course, in Michigan, we have the wildlife reservoir, and this 40% is excluding Michigan. But we haven't been thinking about human to cattle transmission as a potential source. And so that's one of the reasons why we wanted to put these cases together in a paper and start discussing this on a broader level. Next. So the 3 cases that I just reported do show evidence of human to cattle transmission of species. Next. But multiple reports of human to cattle transmission have occurred from around the world. Next. So a One Health approach is needed to address this issue. And in that respect, we have teamed up with the National Milk Producers Federation and have a working group that includes animal health, public health, industry, to talk about best practices, how we can approach this issue from the animal health, public health, and industry to try to reduce the transmission, both of cattle to cattle transmission, human to cattle transmission, and cattle to human transmission of MTB species. Next. I guess we're taking questions at the end. So thank you for the opportunity to discuss this today, and I'll be happy to answer questions at the end. Thank you.
Thank you. Next slide, please. Our next presentation, Experimental Infection of North American Wildlife with SARS-CoV-2, is by Dr. Sonia M. Hernandez. Please begin when you're ready.
Thank you very much for the invite. Next. As we all know by now, coronaviridae is a well-recognized family of viruses that has a wide range of hosts that extend across a variety of different animals. Most virulent coronaviruses in humans are beta coronaviruses, and the ones that are most famous are SARS Classic and MERS, both of which have been due to spillover from wildlife. Coronaviruses have that distinct shape that give it their name, and that's because of the spike protein on the surface responsible for binding and entry into cells that allows it to attach to the ACE2 receptor, which is found in both respiratory and gastrointestinal tissues, which is why infections can result in a myriad of clinical signs. Next. Animals can play a role in the epidemiology of the current pandemic, and we study them for a variety of reasons. Number one, to determine their utility as animal models for human health, or to determine if they can become reservoirs for human health and help us predict future zoonotic and anthrozoonotic events, but also because we care about what the virus might do to individual animals or their populations. And a big part of understanding their role is conducting susceptibility trials. In fact, prior to the current pandemic, several species were already known to become infected with and shed SARS Classic, most notably domestic cats, non-human primates, and domestic ferrets. So in North America, we were already paying attention to mustelids and felids. Next. Several susceptibility trials have already been undertaken in a variety of different critters like dogs, cats, ferrets, hamsters, several species of non-human primates, rabbits, cattle, pigs, and fruit bats. And these have been shown to be susceptible with SARS-CoV-2. And experimentally infected cats, hamsters, and ferrets can also transmit the virus. Specifically about wildlife, Chinese tree shrews have been used as animal models for a while because of their taxonomically closely related relationship to non-human primates. And they were tested by being nasally inoculated with SARS-CoV-2 and found to become infected and shed and develop lung lesions. Raccoon dogs, which were once considered an incidental host for SARS Classic, was also evaluated recently. And Furling et al. Demonstrated that they were susceptible, they shed the virus readily, and can transmit the virus to contact animals without developing clinical disease. And in North America, Griffin et al. Used a captive colony of deer mice to intranasally inoculate them and found that they also could become infected and shed virus and could also transmit to naive deer mice through direct contact without clinical signs of disease. Next. Most recently, Palmer et al., which actually now appears in the Journal of Virology, used fawns to experimentally test their susceptibility, and the white-tailed deer do become infected, shed through nasal secretions, and can transmit to contact naive animals and seroconvert without developing clinical disease. Next, please. So back in February 2020, we started to think about the various ways that we interact with wildlife and the consequences that that might have during this pandemic. So if you'll follow along in this picture, wildlife in the center can come in contact with humans directly through, for example, research activities or through contact in a wildlife rehab hospital, but more commonly perhaps can come in contact with virus shed by humans through waste. And that has been documented by detecting virus in human waste that mirrors the viral load in communities, but also through trash. And then once they're infected on the right side of the figure, that can impact their populations, or theoretically they can come in contact with more endangered species of conservation concern, or once again may transmit the virus back to people. Next. So the objective of our study was to determine if two common North American native wildlife species, raccoons and striped skunks, were susceptible to infection to SARS-CoV-2. Both of these species are found throughout most of the US. They're abundant, they're generalists, they populate urban and rural environments. They commonly present to rehab centers, and they come in contact with human garbage and domestic animals like feral cats. And they reproduce in the spring, which means that at the time that the pandemic was picking up speed in North America, they had high population densities. Next, please. Before going any further, it's important to mention that this work was conducted in a BSL-3 facility. The Animal Health and Research Center at UGA is one of the only facilities in the US that has agricultural BSL-3 rooms, which means that the entire room is considered primary containment and requires extensive HEPA filtration and pressure gradients that dictate airflow. When the researchers are in this room, they have to wear full PPE over scrubs, including Tyvek and aprons and 2 layers of gloves and a personal air filtration unit, a variety of other things, none of which can be punctured or torn by animals because that will not protect them. And this PPE does limit their vision and dexterity. And to make matters a little more complicated, we were experiencing a global PPE shortage at the time of this experiment, all of which had some influence on our experimental design and the number of times a researcher could enter a room. Next, please. We acquired 16 raccoons and 16 skunks, juveniles, from a captive breeding facility, and they were transported to UGA and considered to be healthy at arrival. Next, please. And they were housed in the way that you see here on the right. So the main room contained 2 areas with 2 sets of cages, and those cages contained either a pair of animals each. The orange animals are the directly inoculated animals, and then they were joined by direct contact animals, and that was on the right-hand side, the high dose group, and then the low dose group is on the left-hand side. But note that the airflow in this room does not recirculate. Next. And was unidirectional, and was unidirectional, moving away from the low dose group. The control rooms were held in a separate room under the same experimental design. Next. All right, we fully anesthetized the animals to inoculate them using 2 different doses of a human isolate. The 10 to the 6th dose is what's used in standard experimental trials with this virus, and the 10 to the 3 was meant to sort of mimic what an animal might come in contact with if it consumed, for example, garbage. Next, please. And now I'll give you a brief overview of our sampling schedule. So here we inoculate the animals on the directly inoculated animals on DPI 0. Those are the red spots. And then the next day, it would be DPI 1. Next, please. Then to test for direct transmission on DPI2, we introduced one direct contact to each pair of the DI animals. Those are in orange. Next, please. We collected blood from all the animals for serology prior to the inoculation, DPI5, DPI9, and immediately before euthanasia. Next, please. We collected oral and rectal swabs from all the animals daily until DPI5, and then every other day until the animals were euthanized. We also swabbed food and water bowls on these days. Next, please. And to maximize the probability of detecting lesions during the early stages of infection, we euthanized animals at various intervals. And we also euthanized controlled animals at those intervals for reference. Next, please. And you can, next, please. And you can also play the video. And we sedated the animals to be able to collect swabs. And you can see here an animal that's sedated, that's being prepared for collecting a nasal swab. Those swabs were used for virus isolation and RT-PCR. The blood was used for serology, and after euthanasia, the tissues were collected for histopathology, immunohistochemistry, and virus isolation. Next, please. No clinical disease developed in either of the species or any of the animals of both species, and neither were gross lesions visible. However, we did find that 5 out of the 8 directly inoculated skunks had occasional chronic and mild pneumonia deemed representative of recovery. Next, please. During the virus isolation, Vero E6 lines that you can see in figure A next were used to evaluate each nasal and rectal sample for cytopathic effects, as you can demonstrate in figure B. And positive virus isolation results were verified by RT-PCR. And you can see in the schematic that I have up there, the relevant virus isolation results. And that's that 2 skunks shed on DPI 1 and 2, that's one animal, and then another separate animal on DPI 3, 4, and 5. We were unable to isolate virus from skunk tissue samples, raccoon swabs, or raccoon tissue samples. Next, please. We quantified the amount of virus shed by the 2 positive skunks using plaque assays, and the highest amount shed by a skunk was 2,000 plaque forming units on DPI 4. Next, please. And our 2 PCR results for the nasal swabs are on the table on the right. Only 3 raccoon nasal swabs were positive, but those were all right after the direct inoculation, and it's likely residual virus from the inoculation. Whereas for the skunks, you can see that viral RNA was found up to DPI5 in the skunks. Only 1 skunk nasal turbinate tissue sample was positive on PCR, and we did not detect any RNA in any of the environmental swabs. Now, seroconversion was determined using microtitration serum neutralization assay, and we defined it as a 4-fold increase in titers, and all directly inoculated skunks and raccoons seroconverted. None of the direct contact or control animals seroconverted by the end of the study. Next, please. So what does all this mean? The results of this study show that skunks can become infected and shed a low amount of SARS-CoV-2. Raccoons do not shed, but both species seroconverted. So putting all this together, it's important to note that there were no direct contact animals that became infected from directly inoculated animals. So because we found low viruses from skunks, no virus from raccoons, and really no direct contact animal transmission, it's unlikely that these 2 species are competent hosts for SARS-CoV-2, which is really good news. As for an abundance of caution, we still would recommend using PPE for those people that handle them under unique circumstances like close contact in wildlife rehabilitation. One caveat is that this was a short-term study. We didn't have an opportunity to see how long the animals maintained antibody titers. And I think it is important to mention that knowing that these animals seroconverted under these circumstances needs to be interpreted carefully in future wildlife studies where all that's done is antibody surveillance. And then next, please. A very recent published article in EID by our Colorado colleagues found that deer mice, skunks, and bushy-tailed wood rats became infected and shed virus. But in contrast, a variety of other mammals, which I list up here on the slide, did not, including raccoons. There was one notable difference between our study and theirs, and that's that they did not find that raccoons seroconverted. There are some significant differences in our methodology, sample size, et cetera. And so I direct the readers to this paper, and ours is in the final stages of being published in Frontiers in Veterinary Science for the details. I would like to mention both studies were conducted at the beginning of the pandemic, and thus much more work needs to be done to understand how different viral strains may change the story with these studies. And thank you very much, and I'll entertain questions at the very end.
Thank you. Next slide, please. Our final presentation, The Importance of Plant Health to One Health, is by Dr. David Rizzo. Please begin when you're ready.
Okay, thank you very much. It's a great pleasure to be here. My talk's going to be quite different from the others, and it's going to be sort of more of an editorial on why people who work in One Health should consider plant health as a really important part of that. Next slide, please. So even though the CDC's definition of One Health now includes plant health, as do a few other, the majority of times that we see One Health discussed, whether it's in the classroom or in articles, is something like this figure here, where plant health is generally lumped in underneath environmental health and is not considered often separate from animal health and human health. Next slide, please. So this is the article that figure came from, and I didn't want to just kind of dump on them because I did find their conclusion really interesting in that they say the issue of plant health as a full component of One Health concept is a challenge to be urgently resolved. In fact, human health and animal health are directly or indirectly dependent on plant health. Next slide. And then their final part was raising the concept of One Health to a realization requires also access to a good plant health through productive and sustainable agriculture, recognizing again the potential importance of plant health to One Health. Next slide, please. The United Nations, the FAO, recognizes, and so many of you may know that the year 2020 was considered the International Year of Plant Health. There was a lot of events around this, many of which unfortunately had to be canceled, but it was recognizing this really important concept of plant health. Next slide, please. And the United Nations put out a whole bunch of facts and literature. I'm not going to read all of these. I mean, the key thing to point out here is, plants make up the vast majority of the food that we eat, either directly or indirectly, that plant-based agriculture is a huge part of the economy of many countries, particularly low-income countries, and that the health of plants is under threat from a variety of different directions. Next slide. I like to show this slide to my students, and it really gets home how much plants are involved in our diet. So this particular slide, the more green areas are plant-based agriculture that's primarily consumed by people. The purple are plants grown primarily for animal feed or for biofuels. And you can see certain parts of the world, much of the plants that are grown are for animals. Next slide. I also don't want to forget about natural ecosystems. And, we just heard about zoonotic diseases. So, where many of these animals are living, but also for vectors of endemic diseases such as malaria. So the health of these ecosystems, which are based around plants, is vitally important. Next slide. So how does plant health fit into One Health? And there's a bunch of different ways of looking at this. We can look at food security, so the availability of food, whether it's the loss of crops or the reduction of yield. We can also look at food safety, so food that can actually make people sick through human or animal pathogens. We can also think of plant health as really important, again, as I mentioned earlier, about economics. The picture in the lower right is of coffee rust. So this is not a staple crop, but it's a major source of income for small farmers, particularly in Central America, where this disease coffee rust has really had a major impact. And then again, for ecosystem health, where we see the loss of ecosystems, not just from people cutting down trees and removing forests, but also through diseases, insects, and other impacts. Next slide, please. So when we think of plant health, we just heard about viruses, bacteria, many of the same things that impact people, animals also impact plants. When we look at biotic agents, we see pathogens, we see insects, we see parasitic plants and weeds. In the case of pathogens, you see many of the same ones, bacteria, viruses. Fungi are probably the most common and most important in a broad sense of the plant pathogens, but of wide variety of areas. And of course, abiotic, that many of these are now going to be tied to climate change, water, either not enough or too much. We can also look at temperatures and nutrients, often from cropping the same plants in the same spot for many years. I'm primarily going to focus on the biotic agents, particularly pathogens, in the rest of my talk, but I don't want to minimize the importance of abiotic interactions. Next slide, please. So we can, just as an example of the impacts just of pathogens and pests, this is the production of some of the most important staple crops around the world. And the amount is not necessarily important, but next slide, please. I want to show you what the yield loss to pathogens, to insects are. And you can see with each of these, that a significant part of the crop that's grown each year is lost to pathogens, insect pests. And this doesn't even include abiotic impacts such as drought or high temperatures. Next slide, please. And we can see variation across the world. So you can look on the far left, there's the United States and Canada, in the middle of sub-Saharan Africa, and then in India. And you could see even in a high income countries like the United States and Canada, significant proportions of crops are lost each year. And in some particularly low income countries, up to and over 40% of all crops are lost each year. Next slide, please. So just some quick examples as we go through these. And I'm not going to read all the words on each of these slides, but as I mentioned before, we could talk about food security or food availability. And these are losses due to plant pathogens, pathogens that infect the plant, either reduce yield through just simply making the plant smaller, killing off portions of the plant, or killing plants altogether. And this can lead to losses of staple crops, higher crop prices for people, loss of incomes. Next slide. We can also look at microbes that colonize plants that are not necessarily pathogens, although some of them are, but that these microbes produce toxins that impact people. So the previous slide were pathogens that impact plants. These are plant-based microbes that toxins then impact people and animals. Mycotoxins being probably the most well-known and most important, particularly aflatoxins. And many of these will colonize plants in the field, but also in storage. Next slide, please. Then we can look at pathogens, human pathogens that are associated with plants. They don't infect the plant. They're not getting their nutrient from plants, but they kind of are hitchhiking along. And these are really important. We saw some examples Casey showed at the very beginning, probably in an typical year, up to 50% or more of all outbreaks, food safety, are associated with plants, but these are often also associated in the long run with animals as well, whether they're animal agriculture or wildlife. Next slide, please. And in the case now, we're seeing through the whole food supply chain, so much of our food is coming from all over the place around the world. And this particular one, looking at where a typical salad you might buy, where the food actually came from, the plants. Next slide. And finally, pesticides. Pesticides are incredibly important for keeping yields up. So without them, we'd have even higher losses. However, we do see issues where they're abused, and we see some of these cases often in low-income countries, but also in rich countries like the United States, where we see the impacts of these pesticides. Next slide, please. And all of us know about antimicrobial resistance, and we're seeing this sort of thing with plants as well, particularly with fungicides such as azoles, which are overused on plants, which may then have impacts on people and animals, for example, with aspergillus diseases. Next slide. So how do we tie all these things together in One Health approaches? You know, these ideas of lots of coordination between different groups to really solve some of these big problems. Next slide. Within plant pathology, within plant health, this has often been known as integrated pest management, that we can't isolate pathogens, insects, without considering the entire crop. And so this is, again, focused on plant health, but this idea of working together has long been important in plant health. Next slide, please. And you can see this figure here is really similar to that One Health slide I showed at the beginning of my presentation, trying to bring all sorts of aspects to solving these problems: cultural, host, biological, behavioral, looking at consumers, looking at producers, etc. Next slide, please. And here's one I just found, that I've used in teaching a lot with Nipah virus, where we look at these zoonotic diseases, but you can see pictures of plants in there, the agriculture, and understanding the connection of plants to the whole system is really important. And the plant health aspects and the agriculture is still really important. Next slide. The final thing I just wanted to quickly bring up is this idea of what we can learn looking at human, animal, plant diseases. Next slide. And so the concept of potentially using plants as model systems in infectious disease research, and what can veterinarians, what can public health practitioners learn from things that we do in plant health. And there's a lot of different things that really cut across, whether it's studying innate immunity through epidemiology. Plant pathologists have long had experience, for example, with aerially transmitted pathogens. So there's a lot of examples there, some that are not going to cut across very well, but I think there's many more opportunities here. Next slide. Right, I don't want to ramble on too much. I want to have time for questions for all three speakers, but I hope I've gotten you at least thinking about the importance of plant health as One Health. I could ramble on for a long time with many different examples, but these were just a few examples that I wanted to bring up. And one final slide. I just wanted to thank the folks at the National Academy of Medicine and the Forum on Microbial Threats, where they've really taken in some of us plant health practitioners and started to really look across all of these different areas. So thank you very much. And I guess we're going to move to questions now.
Thank you, and thanks to all of today's speakers for their informative presentations. We have time for a few questions, and we do have a lot of questions for our presenters today. As a reminder, please use the Q&A feature in Zoom if you have questions, and please include the presenter's name or topic. So we have a first question for Dr. Lombard. Has USDA considered recommending to dairy operators that they require pre-employment and regular ongoing testing for TB infection for their employees?
Yeah, thank you for that question. That has come up in discussion as a recommendation, and I think the question is how do we implement that? Because we have had herds that wanted to test employees after they were detected with TB, and they ran into some hurdles on the public health side. So we're trying to come up with ways to get over those hurdles. And also, if a farm is testing their employees and finds an infected employee, what does that mean in terms of the animal health and the quarantine? And so we want to work through some of those issues that might come up, and so we can kind of provide best guidance on how to proceed in those cases.
Thank you. And another question for you, Dr. Lombard. How do you suggest improved collaboration among animal and human TB programs?
Well, I think the best collaboration that first of all, I think that we need to work together and understand where animal health is coming from and where public health is coming from. And there's definitely some limitations on, for instance, on the public health side, what information they can share with animal health. Luckily, in the two herds I presented, public health were able to get those people that were infected to allow them to talk to the herds that were infected, so that we could evaluate or talk to the herds that they worked for, so that they could be tested. So that kind of communication is helpful. I think the working group that I mentioned, where we do have public health and animal health kind of working together to try to figure out where our space is and how we can better work together, is the best path forward.
Great, thank you. And here's a question for Dr. Hernandez. Given that species susceptibility appears to change with the new variants, do you plan to redo your study with any of the new variants?
That's a really good question. If we had the resources, we probably would consider it. We're not sure if we're going to be able to have the resources for that. But I think it's work that needs to be done.
Great, thank you. And there was also another question for you, Dr. Hernandez. You mentioned the methodological differences between studies by your team and Bosco and Louth et al. Might be responsible for differences in the seroconversion in raccoons. Do you have an idea of what in particular might account for that difference?
Yeah, I don't want to get too picky about the details of the two studies, and I think that you do have to kind of go through the papers carefully. I think one big difference is, there is quite a lot of individual variation in titers. And so when you have more animals, you can detect those individual variations. And when you have not as many animals, that's not as apparent. They also did use, different isolate and a few other things that I think might make some difference. This is not meant to be a criticism. It's just the way things go. And that's why we repeat science.
Great, thank you. And we do have a few questions for Dr. Rizzo. One asking if you would comment on the interactions between plants, pollinators, and pesticides.
Yeah, that's a great question. It's a little bit out of my general area of expertise, but I think it falls into that area that, we talk about IPM, the integrated pest management. I mean, out here in California, for example, almonds are the #1 crop for pollinators. I mean, and so coming up with how different pesticides are used, not just locally on individual farms, but cutting across the landscape. And I think that's the key thing with all of these, kind of like the public health areas that, we see with human diseases, that we have to consider the entire landscape. And more I see people working, bringing social aspects into pesticide use to prevent things like the pollinators. And I think that's a really, really important topic.
Thank you. And another question for you, Dr. Rizzo. Are there any resources that our participants can go to learn more about your general field and the role of plants in One Health?
Ooh, that's a good question. I did send a paper we just published in One Health Outlook, kind of a broader theme with what we just talked about here. I think one area that you can see in just about every state is to go to the cooperative extension areas in the local land grant universities for each state. They often have a lot of information on these different diseases, and insects and other things, and how they are controlled. And that's kind of like an entry level part that I would start at.
Great, thank you. And then back to Dr. Lombard. When a herd is depopulated and a new herd is moved in, what would lead the investigation to think the owner infected the new herd versus a failure of cleaning and disinfection once the infected herd was removed?
That's a good question. Like I say, those two studies that reported that were from the 40s and the 60s, and they didn't really go into great detail on what they did in terms of environmental contamination. That was just what was proposed by the authors of those papers.
Thanks. And we have time for one more question for Dr. Rizzo. How do you envision the role of plant scientists in a public health team, and what are some plausible next steps that can be taken towards realizing that vision?
Ooh, big question for the last minute here. So I think in some of these areas, it's recognizing how the plants fit specifically into a problem that needs to be solved. I mean, probably the biggest one that I see, was that mycotoxin example, particularly in areas in sub-Saharan Africa and parts of Asia, that is going to require, interactions between plant scientists, sociologists, because in some cases it leads to how food is stored, which crops are grown. And so all of these things are tied in together. In the article I mentioned before, we talk about a banana disease, which again links to more economics, but it's also a staple crop. And all of these things, animal agriculture ties in with plant agriculture, but then also human health. And so I think all of these, just recognizing that all of these areas are important and then being able to link up with plant scientists, I think is going to be important in the future.
Thank you so much. And we are at the top of the hour. So if you have other questions for today's presenters, or we didn't get to your question, we have included their email addresses on this slide on the ZOHU Call webpage for today's webinar and in today's email newsletter. A video of today's webinar will be posted within 30 days. Next slide, please. And please join us for the next ZOHU Call on September 1st. Thank you for your participation. This ends today's webinar.